Okay, so we're now going to talk about the process of translation. That's how a piece of mRNA, so we've got a piece of mRNA there, how the base sequence in that is somehow translated into a protein with a specific sequence of amino acids. So let's just go through the basics before we look at the details. So now we said that 3 bases on a piece of mRNA, 3 bases in the DNA or on the mRNA code for 1 amino acid. So let's imagine we have on this piece of mRNA, we have U, U, U. Now, in the cytoplasm of the eukaryotic or prokaryotic cell, you have these small molecules here which are called transfer RNA, tRNA. So the black is made up of an RNA molecule, this black outline, and at the end of it, it has an amino acid. Now the one I've drawn there is phenylalanine, and that matches up to this 3 base sequence here, which is AAA. Now that is called the anticodon. Right? And we can see a different amino acid there. We've got glycine, and it will have a different 3 base sequence there. CT, that's the anticodon for glycine. So we have these transfer RNA molecules, piece of RNA in a loop like that with an anticodon at one end and the amino acid at the other end. So let's imagine that sequence UUU, which I've drawn over here. Now, what's going to happen is the only transfer RNA that can hydrogen bond to that is this phenylalanine one. So if I draw, that's going to A will hydrogen bond to U, and so here is our transfer RNA. So there's hydrogen bonding there, held in the right place, and of course it's got phenylalanine stuck to the top end of it, covalently bonded to it. Now imagine that next to it we have another sequence of bases there. We have G, C, A. Now that will hydrogen bond to this anticodon. So there you're going to get this transfer RNA molecule is going to bind, and you've got C, G, T there, and you'll have the amino acid glycine there. Now, this sequence of these 3 sequences, these are referred to as codons, and of course they hydrogen bond to the anticodon, to the correct anticodons. So I've only drawn 2 codons which will code for 2 amino acids, and of course what's going to happen is you're going to form a peptide linkage between phenylalanine and glycine there, and if it's much longer, then you're going to form the whole protein. So that's the basis of it. So in the cytoplasm, you have got transfer RNAs for every single amino acid that occur in proteins. So there's 20 different amino acids. As we'll see later, there actually means there's rather more than 20 different types of tRNA. Right. So where does this process of translation occur? Well, it occurs on the ribosome. So I'm going to draw a ribosome here. Ribosome is made up of 2 subunits. The ribosome in prokaryotes is a little bit different to the ribosome in eukaryotes. And interestingly, the ribosome which you find in mitochondria and in chloroplasts, that's much more similar to the prokaryotic type of ribosome rather than the eukaryotic ones, showing that once more that mitochondria and chloroplasts probably arose by an association with a prokaryote inside a eukaryotic cell. Okay, so the the piece of mRNA is going to line up there. Now, in order for the translation to start, the mRNA must contain a start codon. and the start codon is actually AUG. And it also codes for the amino acid methionine. So that's the transfer RNA. So the transfer RNA, the anticodon will be U, A, and C, and you'll have the amino acid methionine there attached to it. Okay, it all starts with that. Now, I've drawn there, let's pretend there on that sequence, that is where the start codon is, and that's where the mRNA binds to the ribosome. It can't bind anywhere else to start off with. You may notice here there's a bit of mRNA before the start codon, and that will be there, but it will, of course, it won't be translated. Right, once the piece of mRNA has bound to the ribosome is actually made up of RNA itself, ribosomal RNA, and quite a few proteins as well. It's actually big enough to be seen with the electron microscope. People could see them before they actually knew what they did, and it's like a multi-enzyme complex. And interestingly, some of the RNA actually, as well as the proteins having catalytic properties, enzymes which you're familiar with, some of the RNA molecules there have catalytic properties as well. Once the RNA is bound to the ribosome, then what will happen first of all is you'll get the methionine tRNA. We'll draw that with a will bind there, and then you'll get a whole, and all the other tRNAs, the anticodon will hydrogen bond with the codon, and they're all going to start lining up on the mRNA. I'll draw them different colors to show the different amino acids. And what happens is the ribosome actually moves down the piece of the mRNA, and as it goes, it joins these amino acids together to form the protein. So the protein chain will start growing that way. So if you could imagine a ribosome here, there's a ribosome, and here is a protein chain which is being made. Just drawing the amino acids all in black there. There's the protein chain coming over, and as the protein chain comes off the ribosome, it folds, and this is the piece of mRNA, which is like a piece of ticker tape going through the ribosome, and the ribosome is moving along in that direction. As it moves, this line, this polypeptide, gets longer and longer. Okay, that's protein synthesis then. So in eukaryotes, that's going to occur in the cytoplasm, and also the ribosomes are quite often bound to the membranes of the rough endoplasmic reticulum. In prokaryotes, it's just going to occur in the cytoplasm. So let's just talk about this genetic code. So very briefly, we're not going to go into a great deal of detail, but if you have 4 bases and you have them in triplets, that means there are a total of 64 codons altogether. Codons. Of course, we know that means there are only 20 amino acids. So some amino acids have several different codons, and so the code is said to be degenerate. Now, let's just have every amino acid actually has more than one codon, but let's just have a look at for example, glycine. Okay, glycine, the simplest amino acid, it has the following possible codons. It has, where are we? Right, okay, so it can have GGA, GGU, GGC, and GGG. Now this is a pattern which occurs quite frequently in the genetic code. The last one here, so anything beginning with GG it's got to be glycine, and the last one doesn't really make any difference. That also occurs for the amino acid alanine, which is it's quite a simple amino acid. In the case of alanine, it's GC, starts off with GC, then it can either be A, U, C, or G, and they all give you alanine. That's quite a common theme throughout this. Other amino acids only maybe have 2 codons. So cysteine, which is one which is important for forming the disulfide bridges. Cysteine. There's only 2 codons for cysteine, but they're pretty similar. They both start off with UG, and the second letter, the last letter, sorry, can be C or U. So cysteine has 2. So you get a rough idea of this degenerate code. And what's also pretty important is we've talked about the start codon, which is AUG. And that also codes for the amino acid methionine. So does that mean every single protein starts off with methionine? No, it doesn't, because in some proteins, the methionine which is put there is kind of chopped off as part of the processing of the protein before it's matured. The first methionine goes. Also, to stop the process, you need a special codon to stop the process, and there are 3 different stop codons. The 3 stop codons are, you can have they both start off with UA. So you can have UAA and UAG. And there's a third one as well, which is UGA. So whenever you get any of those codons, what happens is on the ribosome up here, the mRNA and the protein detach, and that is the end of translation. Needless to say, it can be quite damaging if you get a mutation where a codon, so for example, let's think of if you had the codon UAC, so you have UAC, if that got mutated, just the last letter got mutated to UAA, so all that would have to happen on the DNA would be that a C would have to be changed for an A. If that happened, instead of having this codes for the amino acid tyrosine, this codes for stop. So if tyrosine was in the middle of the protein, instead of getting the full length protein, you'd get a short protein, which obviously wouldn't work, and so it would lead to a non-functional protein. So that could potentially be a very damaging mutation, one that the cell may not be viable. Okay, so there's a little bit about protein synthesis and translation of messenger RNAs.